DNA's big mix-and-match party. What happens when foreign DNA crashes yeast's genomic bash? In a recent paper published on February 7, 2025, in Science, Leah Meneu and her team from the Institut Pasteur have stepped into an adventure straight out of a sci-fi movie, only with more yeast and less alien lifeforms. Their study, titled Sequence-Dependent Activity and Compartmentalization of Foreign DNA in a Eukaryotic Nucleus, explores what happens when foreign DNA decides to join the party in budding yeast, scientifically known as Saccharomyces cerevisiae. The core of the research reads like a genomic soap opera. When foreign DNA, specifically from bacteria or other eukaryotes, is introduced into a yeast nucleus, the DNA doesn't just blend in like a quiet guest at a dinner party. Instead, it behaves based on its GC content. Think of GC as the amount of geeky and coolness factor your DNA has. If the foreign DNA's GC content matches the yeast's, it's like finding a friend with the same taste in music. They laugh, swap stories, and generally hit it off. The DNA intermingles and becomes active, singing along with yeast's genetic code. Conversely, if the GC content is off, it's more like your cranky uncle who's tending toward a secluded corner, serving as a silent blob, separate from the jovial genomic festivities. This study doesn't just stop at mapping this introvert-extrovert DNA behavior. The researchers discovered that these foreign DNA sequences spontaneously form distinct nuclear compartments. Large DNA strands with more geeky GC content become the social butterflies, actively transcribed and mixing with their host's chromosomes. Meanwhile, the ones with less GC content are like the guest who doesn't mingle much, forming a separate, inactive compartment, a blob of chromosomes off to the side. But what's underpinning this social dynamic? The researchers used yeast as a high school prom to observe DNA mixing, and found that active transcription in the host genome is key. This mixing is predicted accurately by the DNA sequence, making chromatin's social behavior more akin to an organized dance than one might expect. Challenges and opportunities. While the assemblage of yeast and foreign DNA is intriguing, it's not without challenges. For instance, the study notes that transcription of these foreign sequences sometimes follows their home orientation, and not always harmoniously. This research showcases genetic predictability, but also hints at unanswered questions. Can these findings translate to more complex organisms? How do these principles hold up under different cellular conditions? On the opportunity front, this genomic soiree opens doors to fascinating future applications. The deterministic sequence-based DNA behavior observed here could aid synthetic biologists in designing more predictable genomes, paving the way for customizing yeast strains or other eukaryotic organisms in biotechnological applications. Outlook. Looking ahead, this research could drive advances in genome editing technologies and synthetic biology. By understanding the DNA party rules, scientists could engineer microbes with highly specialized functions, helping in everything from sustainable biofuel production to medical therapeutics. Imagine your DNA sequences engineered to play along nicely, much like a perfectly curated mixtape at a party's, ensuring everything syncs and sways to the right tunes. For those interested in all the technical goings-on, the full study is worth a read. But for anyone who's ever wondered what happens when foreign DNA pops into a yeast cell, it's clear. The nucleus transforms into a social gathering, echoing the importance of genetic camaraderie and the universal pursuit of a balanced dance between foreign and familiar.